you're after. Weighs 200 grams. I felt very embarrassed about winning the bloody thing. So I gave it away as quickly as possible. I'd actually heard that some people had their Nobel Prizes stolen. Well, I mean, what do you do with the bloody thing, right? I mean, there it is. I keep quite a lot of these. Once somebody said, well, I mean, my advice would be to, if you're going to win one prize, go for the big one, all right? Because you keep on getting stuff with it. There's an awful lot of, people just then give you medals and things after that. So I have a downstairs lavatory is just full of these plaques and medals and things all piled up, but not the prize. So I gave that away to my college in Cambridge because it was the tradition, if you were a fellow of this college, you had to give them some silver. Of course, if the silver was gold, that was even better. And they had a huge safe with an enormously thick door, and somebody to polish the silver. You don't have to polish gold. So that was, anyway. So, I think it's worth saying, it's worth pointing out that actually, I think wanting to win a Nobel Prize, which I suppose I did, is a completely admirable and perfectly legitimate thing. Because if you're a scientist, surely, you would like to solve the most important and interesting problem you possibly could. What's wrong with that? I mean, that's what I think we all ought to be doing, right? But, as you'll see, you have to be fantastically lucky in order. I mean, I don't think that I'm a fantastically good scientist. So we'll put it this way, I know a lot of scientists who are a lot better than I am. And they're never going to win the prize because they never had the luck of actually being in a place where suddenly they open the door and there are amazing things behind it. So let me show you how I got that. Yeah, this was the referee's comments on the Nobel Prize winning paper. Wild speculation based on faulty logic. And the other reviewer was Paul Nurse, who liked the paper. I don't know who this was, and he or she, the editor overruled them. So that was good. But it's always been my experience, by the way, that if you really discover something new, which is true, that you have a lot of trouble with the reviewers, because people don't like new ideas. So if you have a lot of trouble with reviewers, maybe that's a compliment rather than, it can be bloody irritating, I know. So I grew up in Oxford. We actually moved there at the end of the war in 1945 because my dad, who was a medieval historian and a librarian, got a job in the library, and here is my dad reading some sort of Greek manuscript. I don't know what it is. And he was a man of few words, and I would say I didn't know him all that well, really, because but he was, he had all these friends, and he was a very generous person. In fact, almost all of the books of that generation acknowledge his help. And I like to think that I carried on. So I started my performing career at quite an early age, and I think it was very useful. I mean, after that, I got a real taste for acting and singing and dancing, not dancing, dancing. But we did a lot of Shakespeare and Gilbert and Sullivan and things like that. There was a Schubert opera, Beggar's opera. I mean, I had a not a great voice, but a reasonable voice. So it's fantastically useful, actually. It always gave me great self-confidence. I mean, I'm not by nature a terribly confident person, but acting, entertaining people, it was good. And then I had some wonderful teachers, of whom the first was this man here, Gerd Sommerhoff. He was a young German who had been hired by the first school I went to, and his instructions to the headmaster were to just keep the boys interested. And he did keep the boys interested, and he kept us interested, partly because he was interested in little boys, and that got him into trouble. In fact, it's actually awful. If you look him up on the internet now, you know, that's what you'll find about him. So now it's terribly fashionable to talk about child sexual abuse. Well, he was a child sexual abuser, but he was a fantastically good teacher. So it was he who told me that I was a biologist. Because I just did terribly well in a biology exam when I was about 11. I wasn't particularly interested in biology, but I found that it came very naturally to me. I came out almost sort of 10th in the whole school when I was in the middle of the school. And I realized I'd done no work for this thing. I mean, I didn't revise or anything. It just came naturally. So that was very helpful. So I knew from about the age of 11 that I was going to be a biologist. Which was just as well, because unlike most of the people who have spoken, I've only ever failed 2 exams in my life. One was the driving test, and the other was a O-level advanced maths. I was really bad at maths. I just didn't get it. I mean, even when we did mental arithmetic, I couldn't do it. And I would like to be better at maths, but I'm not. So I did okay. You know, you go on passing exams, and I managed to get into Cambridge, which was great, because actually, though we didn't realize it at the time, the people who go to Cambridge are the best scientists of their generation. We had absolutely no idea about this, but it turned out to be true. And I had some wonderful teachers and also wonderful colleagues. And it was a wonderful time. So this was I was an undergraduate from 1961 to 64. And I remember very well when François Jacob came to give 2 lectures in Cambridge, I think in 1962. And we all went to listen to this. This was the first public explanation of the control of the lap operon. And it was absolutely fantastic, you know, revelatory, the beauty and the clarity of it. So I went back to my biochemistry supervisor and I said, you know, is this true? And he said, well, I don't know, it's a bit early to say. Which proved that actually a lot of people in Cambridge hadn't actually been following this stuff, because a lot of it was written in French. And it looked a bit biochemical, but it was, if you read about it, it was fantastic. Now, the thing is, I'm rather an expert, because I grew up in Oxford among all these medieval historians, and then went to Cambridge. I'm quite an expert on the differences between Oxford and Cambridge, and actually, although people often lump them together, they're very different places. I mean, almost in every particular, they are different. I mean, how people get paid is different. The kind of courses are completely different. So the science course in Cambridge is fantastic, because the rule is that in your first 2 years, you have to do 3 experimental subjects, and then maths. And that forces people to diversify. And almost no university that I know of, apart from Cambridge, has, at least in the UK or the continent, has this arrangement. So you can choose. I mean, I did a very eclectic mix of substrate. I did organic chemistry, invertebrate zoology, physiology, experimental psychology, and biochemistry. And it was a very good mixture, because the physiology, for example, went very well with the invertebrate zoology, which was a comparative physiology course. So we looked at worms and snails and all kinds of weird creatures. And the physiology course was wonderful. I mean, we were constantly ripping the hearts out of rabbits and things like that. watching what made the heart beat. And if you've ever done that experiment, you take the rabbit, you take its heart out, you fuse it with Ringer solution, the heart goes on beating. Then you take the calcium out of the Ringer solution, the heart stops beating. You add the calcium back into the Ringer solution, the heart starts right up again. I mean, you never forget that kind of thing. And then people were breathing their own air over sodium hydroxide, so they passed out when the oxygen concentration. So they were made to do mental arithmetic, you know, the supervisor. When they sort of finally slumped on the desk, he ripped the thing out and allowed them to breathe properly again. It was, or they brought in, you know, we were talking about Huntington's chorea. So a patient with Huntington's chorea was brought up. So it was terrific. Well, my favorite was the cat. So we had these demonstrations, and there was this cat on the, there was a bench in the middle, and there was a cat, and the cat had a ruff around its neck. And the demonstrator came in and said, "Ladies and gentlemen, I would like to assure you that this cat is dead." He pulled the head off the cat. And he then proceeds to sort of stimulate the brainstem of the cat, which then arches its back. It does absolutely everything a cat has done with no head on it. I mean, you know, wow. Well, I mean, now you can't do that kind of stuff nowadays. Boy, it was fun. Or, I mean, how many of you have you seen the pancreatic juices of a calf come out? I mean, you know, I'd never really thought about this, but the duct that makes all the digestive enzymes, you know, this was cannulated. So you could see the enzyme just dripping out of this deceptive, fabulous stuff. I mean, you just never sort of think about what that actually means in practice. So it was very educational, and as I say, it was ready to each other's. So I used to. This was a shortcut to the lab from where I lived, the old Cavendish. And this again is sort of about Cambridge. So you'd walk through there, and when you the other side, the following things had been done within a space of about, you know, the size of this room. You could touch them. Maxwell was the first professor of physics. He worked out his equations, worked out the relationship between electricity and magnetism, and realized that light was an electromagnetic ray. Thomson discovered the electron. Rutherford and his boys had discovered the atomic nucleus, the proton, the neutron had been first to split the atom. Watson and Crick worked out the structure of DNA, and Kendra and Perotz worked out protein structure. So this was, I mean, by any standards, this is a pretty amazing series of very great discoveries of the late 19th and 20th century. So that when you went through that arch, you thought, oh wow, well if you're feeling good about yourself, you go through, oh wow, you know, I'm in this tradition, I can do this. And if you were feeling not so good about yourself, you go, oh Christ, there's no way I could live up to this. The weight of tradition is too more than I can bear. Here I am, a miserable worm. You know what I mean? So, but I think that tradition is actually important. Of course, I was, you know, it was really born with a silver scientific spoon in my mouth, because not only was that thought, but also the fact that, you know, we had lectures from Fred Sanger, from Sidney Brenner, and we had practicals where people from the LMB came down and showed us how they were doing. I mean, we were in the presence of, I mean, you know, one thing that was clear was that these great scientists were a very heterogeneous lot. Some of them were clearly brilliant, some of them didn't look very brilliant. I mean, Fred Sanger always rather looked like a gardener, actually. But it's quite clear, you know, that he was, in many ways, the best of them all. Not many people win 2 Nobel Prizes, and Fred did, you know. He did it by doing things that people at the time thought was impossible. We were told, for example, that it would be impossible ever to sequence DNA. wasn't impossible for Fred, but it took him a long time to do it, you know, but we had contact with those people. And so you kind of knew that, you know, you couldn't get any better than that. And in some cases, I mean, I remember Francis Crick used to come to a lot of seminars and ask questions. And quite often he'd ask questions which I was very surprised about because I knew the answer and he didn't. And it dawned on me that actually Francis only asked questions when he didn't know the answer and he was just seeking clarification. That was a wonderful lesson, actually. But it, you know, you realize that he might be a genius. I mean, Francis was actually the person who really looked up to because he knew everything from physics to biochemistry. But he also didn't know things, so it was okay not to know things. That was a very good lesson, I think. But anyway, I always, because I was sort of good at chemistry and good at biology, always wanted to be a biochemist. Now, I think it was lucky, and that was the time when it became clear that the program, which was really initiated by this guy, I mean, you should read some of the lectures that he gave, saying that we will be able to understand life in terms of simple chemistry and physics. Now, there were a lot of people at the time who didn't believe that. There were a lot of people who actually thought that life had something special, you know, the vital spirit. But not this. He presided over an amazing department. I'll just show you a couple of sort of things. They published. They were very left-wing, very radical. Many of them were communists in the 30s when he was in their heyday. So the question was, you know, I passed my exams and did okay, and decided to do a PhD. So the question was what to work on. So I went to my supervisor and I said, what shall I work on? He said, why don't you go to the library and find yourself a problem? So I went to the library, spent about a week there, and found myself a problem. And the problem involved making rats nephrotic. And if you make a rat nephrotic, that means it raises antibodies against its glomerular membrane, so that it pees out a lot of albumin. You get albuminuria, and the liver is stimulated to make ups its albumin output. So I tried to do this. You had to kill a rat, get out its kidneys, mash them up, and inject them into another rat. Fortunately for me, I used the wrong strain of rat, so it didn't work. And I also discovered I hated killing rats, and the rats hated being killed. So that wasn't very good. So after about, you know, 3 or 4 months of not making the rats nephrotic, I haven't really got a problem. And of course, if you don't have a problem, you've got a problem. So at that point, I went to the first scientific meeting I ever went to, and it was, I must have heard many talks, but these 2 talks made a deep impression on me. And the first one was by Henry Borsuk, and this is his title, Early Development of the Sea Urchin Egg Compared with Erythropoiesis. Now, what do sea urchin eggs and red cells have in common? They are both round. That's it. But Henry Borsuk had worked on both of those problems, and he discovered that when you fertilize sea urchin eggs, protein synthesis turned on. That was interesting. And he'd also discovered that if you starve red cells of iron, protein synthesis turned off. The iron, of course, was necessary for making the heme. And so that was rather good, and you'll see why in a bit. But forget about that. The more important immediate one was this talk by Vernon Ingram, the man who discovered the molecular basis of sickle cell anemia. And Vernon gave a talk, which was also about the question of how and when the heme was added to hemoglobin. So there's half of hemoglobin. That's an alpha-beta dimer with the heme carrying oxygen in it. And what Vernon said was that, so when does the heme join the protein? Well, he presented evidence that what happened was that as the nascent chain came out of the ribosome, it sort of began to fold up. and the heme should be put in about here. And what he said was that the ribosomes waited for the heme. And that's why if you took away the heme, they couldn't get past this block. They formed a roadblock. So we thought, gosh, what an amazing thing. How did the ribosomes know? So the graduate students went back to the lab. Where are the graduate students? Here they are. Tony Hunter and Lou Reichhardt. And discussed this. And we realized that he was talking through his hat. He'd got it completely wrong. If anything, his data showed that it was exactly the opposite way around. So, and I can't quite reconstruct this, but luckily for us, Lou had been making these, no, very distinguished neurobiologist, he'd been making these reticulocytes. He was only there in Cambridge for a year on the same bench as me. And he'd learned how to make reticulocytes. And actually, he was studying this question of whether heme stimulated protein synthesis in an in vitro system. Never got it to work, actually. Tony actually joined us a year later. So there's the hemoglobin with the heme poking out of it. And so we decided we could do the experiments better than Ingram, and let's find out the truth of the matter. So basically what happened, Lou then went back. By the way, he was a very good mountaineer, and he's pointing here at a little piece of the top of Mount Everest that he collected himself without oxygen. So, and I once climbed Mount Snowdon with Lou, my peak of my mountaineering career. And in 1966, I got fascinated by this thing. I mean, the long and short of it was what Tony and I collaborated on our PhD, basically, and found that actually the ribosomes did not form a queue. The ribosomes were actually randomly distributed along the messenger RNA, as far as we could tell. And I went to my second meeting, which was in northern Spain, in Thessaloniki, and ran into this guy here, Irving London, who, unbeknownst to me, was the chairman of the Department of Medicine at the Albert Einstein College of Medicine. And by a sort of funny combination of things that it involved me getting slightly drunk in a Greek taverna at lunchtime and running into Irving when I came out. And he said to me, Tim, so what are you going to do when you finished your PhD? And I said, well, to hell with finishing my PhD. Couldn't I come and work in your lab this summer? And he said, well, why not? So I went and worked in his. I was the first graduate student ever to take sabbatical leave of 4 months. And I loved being in New York. And you know, we worked on this problem. And then I went back there as a postdoc. So there was one interesting thing about the sort of PhD that Tony and I did together, really, which was that we discovered, again accidentally, and again partly because of getting slightly drunk, we let the centrifuge run a bit too long over lunch, went back and analyzed it anyway, and discovered, as a result, because the large polysomes had all run off the bottom, that actually alpha chains were made on much smaller. There were many fewer ribosomes on alpha chain message than on beta chain message, although they were the same length and had the same room for both. Okay? So that was really interesting. That was my first Nature paper. I mean, you didn't have to do much to publish a paper in Nature in those days. But it was kind of interesting. And then what was even more important was that we then did experiments to find out, because there were two possibilities. One, the rate of initiation was different, and second, that the rate of translation was different, okay? And we concluded the rate of translation was different, and the reason why we concluded that was because we failed to do a really elementary control. And Harvey Lodish set us right in print later, and I was very embarrassed. You know, we published this paper which said ribosomes translated alpha chains faster than beta chains. And we got it wrong, because so ever since then I've been very keen on obvious negative controls, because you don't want to be wrong in health. So anyway, I went back, when we'd finished, I went back to Irving's lab, and by that time, some people actually in Cambridge, unbeknownst to us at the time, had worked out that you could make a cell-free system from these these reticulocytes, which responded to heme. And so this is the kind of time along the bottom, counts in protein up the side. Normally, this rate of protein synthesis is very similar, I mean, virtually identical to the rate that they would have been doing in the intact cells. And if you leave out the heme, then protein synthesis curls over and dies. It's about 5 to 10% of the rate. It sort of keeps going, actually, which is probably important. We didn't pay much attention to that. If you add back the heme at this point, then the protein synthesis takes off again. So, and the great thing about this preparation, you pellet the reticulocytes, you simply add water, they break open, you spin off the junk, take the supernatant, freeze it in liquid nitrogen. You can do an experiment any time of the day or night, any week, any month of the year. Incredibly reliable, and the question was, what is the switch that heme controls? Well, what I actually did as a postdoc was to repeat this experiment over and over again, which was incredibly reproducible, but it actually didn't get us much forward in finding out what the mechanism was. But if you do the same experiment over and over again, you keep on getting the same result. You've got to think of some way of asking. And there was really no way of asking, so we did it out. So as a result, I found myself, as a postdoc, at a bit of a loose end. And I had a technician who was about twice my age. She was called Grace Vanderhoff, and she was very, very nice. And when she went home, I started doing secret experiments, because my boss had also decided to go to MIT to found a new medical school, so he wasn't around either. So the minute Grace went home, I started doing experiments. So, you know, in secret at night and at weekends. And I decided that I would map the genes in poliovirus. That was going to be my project, because I realized that I could do it. Except that poliovirus didn't get translated in reticulocytes, as I discovered pretty quickly. So I reasoned that the reason they didn't get translated was that reticulocytes don't get polio, whereas HeLa cells can. So I asked Ellie for some poliovirus-infected cytoplasm, which by definition had the factors that would allow the translation of the poliovirus, added it into the system, and discovered to my amazement that it inhibited protein synthesis, just like the lack of heme. So I thought, gosh, that's amazing. What is this stuff? Well, to cut along, so it turned out the stuff was, it resisted digestion by DNase, by RNase, by proteases. It was heat stable and large. So that was very puzzling for quite a long time. But at this point, Nixon was invading Cambodia, so we used to go down to Washington to get tear gas and protest against. It was very, very funny time. I mean, one felt almost, sort of almost immoral to be doing experiments when such terrible things were happening in the world. We used to have also tremendous arguments over the lunch table because but it turned out in the end we realized that the reason why this stuff was RNase resistant was because it was double-stranded RNA. And then I worked out how you digest double-stranded RNA. You needed microprotyl nuclease, and don't forget to add the calcium. And that was another Nature paper, actually, because we discovered that tiny amounts of double-stranded RNA inhibited protein synthesis in this system. And that was very curious. And the symptoms were always the same, namely, there are the polycystes, the sucrose gradient. One had to do lots and lots of these things, and single ribosomes, dimers, trimers, tetramers, pentamers, and just a hint of a sixmer. And if you inhibited it, they all disappeared. Okay? And the key to what was going on really came from this experiment here, which I didn't do until I came back to Cambridge. But I went back, and I'll show you, I joined actually Tony and my friend Richard Jackson, and we had a sort of, this is another interesting thing, that I went home, I had a college fellowship. I didn't have an EMBO fellowship, it didn't exist in those. There were very few fellowships in those days. But I was sort of going back among friends to the biochemistry department. Richard and Tony had discovered that hemoglobin synthesis, in fact, all of protein synthesis in eukaryotes was initiated with a special methionine tRNA. So this was a real breakthrough. So we started labeling these gradients with methionine. And what we discovered, we're now looking, the polysomes are all right off the right-hand end here. And here are polysomes with radioactively labeled nascent proteins on them. You see this big peak over the 40S subunit. And so well, people had noticed that and said, oh, initiated tRNA, was initiated tRNA, is sticky. But they'd not realized the significance. And then one day we did this experiment, and this is, I think, yes, this is minus heat. That peak disappeared before the polysomes disappeared, which proved to us that this was a precursor to that. And then the question was, you know, did this peak have messenger RNA? And the answer was that it didn't. And that really put the cat among the pigeons. So we realized that the way that protein synthesis is initiated tRNA binds, you have the tRNA there, and then the tRNA, the ribosomes scan along the messenger RNA, seeing whether the anticodon of the initiated tRNA matches the first AUG. And again, we ran into tremendous trouble with the reviewers, you know, who really didn't believe this story, because everybody knew that tRNA only bound to ribosomes when you had message there. And the idea that the tRNA could sometimes bind first, even though it made sense to us anyway, was very controversial. But eventually, with some help from friends at the LNB, we managed to prove that this was the case, and it was the case. And I said, this guy here, Richard Jackson, I sort of ran the lab. I wanted to make this point. So I was very lucky. I mean, I had a college fellowship, and then I got various fellowships, what we call the Bike Fellowship and the Royal Society Fellowship. And I remained a postdoc for about, I guess, from 1971 to about 1982. And I had no responsibilities. Richard had a job, which meant that he couldn't do, you know, he was busy. And so we really ran the lab together, and that was absolutely great. It worked extremely well, and we were very complementary because Richard was a lot cleverer than I was, but he tended to get sort of stuck in minor details, whereas I was always trying to sort of, you know, take broad brushstrokes and get on with stuff. So it was a very, very, you know, it was like this real symbiotic relationship. I mean, you know, you didn't have to explain what the experiment was. You both look at those superimposed gradients, you know exactly what was going on. It was a very, very happy time with some excellent graduate students, I may say. I mean, Hugh Allen, who is now the head of the LNB, was a graduate, sort of joint graduate student between Richard and me, and it was just a joy having Hugh in the lab because he was so good. He was one of those people who was sort of creatively lazy. We were always looking to do things quicker and easier. You know, and Hugh was brilliant at this. You know, did you really have to incubate things for half an hour? What happened? You just left it for 5 minutes. And quite often you find that 5 minutes was quite enough. Thank you very much. Stuff like that. It was terrific. And then the lab burned down. And Richard rang up one Saturday morning, about 7 a.m. and said, Tim, the lab has burnt down. Don't bother to come in because there's nothing left. So of course I sort of put on my, rushed in. And indeed the lab had burned down. But there was the liquid nitrogen duo with our most precious samples in it, which was still venting liquid nitrogen. The seal had melted, but it still had some nitrogen, so we quickly found a new duo filled it with nitrogen, put all the most precious samples. So within about 6 weeks, we were back in business. I remember pH meter was the last thing to arrive. We had a wonderful sob story to tell. So our lab has just burnt down. Could you possibly get us a new centrifuge? And people, the Beckman would say, well, you know, we were going to send 5 centrifuges to AstraZeneca. I'm sure they wouldn't mind if they only had 4, and you can have them. You know, stuff like that. And luckily, we actually, our lab was on level 5 BU5, whatever that is, which is all about where I'm standing. This is the old LNB. And it was fantastic because Max Perutz, who ran the place, said we could use his stores, and more importantly perhaps, have lunch in the tea room. And this was a phenomenal education. Because in that tea room, you know, you'd sit down and Francis Crick would come and explain about nucleosomes. I mean, it was just fantastic. I mostly hung out with the Gurdon lab, actually. I was friends with people like Doug Melton and Ron Lasky and co. And we did, you know, experiments on TMV with one of Eric Klug's graduate students, David Zimmer. And the interesting thing about this is, so I mean, we were in total awe of these people. These are all Nobel laureates. But in our generation, people like Bob Horowitz, John Solston, Andy Marty, Roger, you know, these were all people who were just around. We didn't think of ourselves as genius scientists. We thought of ourselves as, you know, we had interesting problems which we would describe to each other, and on the, you know, it was just a sort of very nourishing kind of atmosphere, actually, because what could you do? I mean, you just had to do what seemed sensible, and you discussed it with people, and they'd tell you if you were being stupid in the nicest possible way, and you know, and really, sort of as a result of that, we very quickly solved our problem. And in a way, you know, we actually, just before the fire, all the graduate students, thank God, had graduated, so there weren't any theses burned up or anything like that. We lost one paper, I think, which we never bothered to do, because this was so much more important. We were totally confused. I mean, a deep fog about how double-stranded RNA and heme control protein synthesis. And suddenly everything came together, and it was really Richard who provided the key clue. And this is sort of really, I think it's quite subtle, but very interesting. Namely, it turned out the control needed ATP. Now, you could never find that out as long as you were using protein synthesis as the assay, because, of course, protein synthesis needs ATP. But we changed the assay subtly so that we looked at an initiated tRNA binding to the 40S subunit assay, and it turned out in order to inhibit that, you not only had to have GTP, which the reaction needed, but to inhibit it, you needed to add ATP. And so once you knew that, it was pretty obvious to just, you know, it was just add this an ATP to the reaction, see what gets phosphorylated. And this protein here got phosphorylated. Luckily, Theo Stalin had recently discussed how initiated tRNA binds to ribosomes. When I say, I mean, it purified this factor. And upstairs, you'll find Candria's wonderful poster, which in 2015, the structure of this complex, which I've shown you, has just been solved. And this is really nice. So here's the initiated tRNA. Actually, you can't see the initiated tRNA. It's somewhere buried inside there. It's always that. And where are you? The green one. The green one. That's a tRNA. Okay, yeah. And that's the factor. And this is the thing that gets phosphorylated. What's interesting, actually, is that the phosphorylation doesn't stop this reaction from going on. It then becomes a one-shot deal. What happens is that you can't get this stuff off if you phosphorylate the AIF2. It gets stuck as a GDP complex, and the thing doesn't work. So that was all rather puzzling, and got fairly lost. So the net upshot of this was that basically, you know, we found this protein got phosphorylated. That was the answer. We found out how it worked. We'd solved the problem that I've been working on for basically the preceding 10 years. So I didn't have a problem anymore. So we organized a meeting about the control, the translational control of protein synthesis. And I thought it would be fun to invite Tom Humphries to that meeting, because he was the only person I could find who seemed still to be working on protein synthesis in sea urchin eggs. And I just wondered, remembering Borsuk's lecture, whether anything interesting, because that was the exact opposite, that when you lack heme, protein synthesis shuts off. When you fertilize a sea urchin, protein synthesis turns on. Could it be the same mechanism in reverse, perhaps? And so the obvious thing to was to invite the expert to this meeting, EMBO meeting we had. And Tom came, and unbeknownst to me, he was a very keen cyclist. And he said, Tim, where could I rent a bicycle? And you couldn't, in those days, rent a bicycle in Cambridge. Nowadays you could. So I said, well, I'm afraid there isn't any way you could rent a bicycle. Why don't you borrow mine? So he went for a bicycle ride. And on the basis of that, we became friends. And what I didn't know was that Tom was the director of the embryology course in Woods Hole. Here is Woods Hole, Massachusetts. You can see it's a lovely place with sailing ships. And the lab is where Ron and I worked, is sort of there. That's the eel pond. And that's where the fancy folk live. It turned out that Tom was running this course. He said, why don't you come and help teach the course? So that was a wonderful opportunity because there were no sea urchins in Cambridge and Tom was the expert. And so I said, great, I will come and teach the course next year in 1977. And so I went in 1977 to teach in the embryology course, knowing no embryology, but that didn't matter. So I lectured on protein synthesis and stuff, and that was okay. But the problem was that the lab was very poorly equipped. In particular, it didn't have any freezers. The guys downstairs, the neurobiologists, did have some freezers, but they were really snarky about letting embryologists share their freezers, so that wasn't really a viable option. So I vowed I would never go back there again, ever in my life, because it was just hopeless. And all we did was eat pizza, go to the bar, and dance in the evenings. We also did, I mean, it was very good, a lot of discussions, a lot of scientific discussions, and I met a guy called Stanley Cohen, who was to be a Nobel laureate, and who had discovered epidermal growth factor, and Stan and I got on very well and played tennis together and go bike rides together, and that was nice, and he was a fantastic scientist, and I learned a lot from him. But I found the following year I didn't go back, and then I missed the place terribly. And when I went back in 1979, there's Tom, who died last year, sadly. There's Joan Ruderman, who became the director of the whole lab, and there's my friend Eric, who's Joan's graduate student, with whom I got on very well. That guy there is Matt Winkler. He became a millionaire selling RNA, believe it or not. And I came with a suitcase full of Eppendorf tubes and tips and things, because I knew what was missing. So, you know, and gel apparatus, power packs and things, you know, just little things you need for running the lab. And that was fine. And we made progress. And getting sea urchin eggs was pretty easy. You give them a 12-volt alternating current electric shock. And if they're female, they shed their eggs like that, and they sink to the bottom of the beaker. If they're a male sea urchin, they do that, and that's the semen. And if you mix the two together, it was easy to confirm Borsup's finding unfertilized eggs hardly make any protein at all. You fertilize them, and after a lag of 5 to 10 minutes, protein synthesis takes off. That's naturally my own repetition of Borsup's experiment. And so, you know, we started looking, and we decided to look, see if things got phosphorylated, and as far as we could tell, well, actually one protein did, and that it wasn't the answer. I mean, the net result of this was that we worked and worked and worked on this, and in that summer Matt discovered that actually it was the change in pH that turned on protein synthesis, but we never did discover how. So I went back year after year, and I switched from being in the embryology course to the physiology course, which was a much more natural home for me. But one other thing happened in that summer of '79. Yeah, and sorry, getting ahead of myself. When you look down the microscope, which I learned how to do, because I'd never done that before, and if we add some sperm, not only does protein synthesis turn on, but the eggs start dividing, and what you can see is they divide incredibly synchronously, and that synchronous division is completely natural. You don't have to do anything to make the division synchronous. They just are, as long as you add the sperm and mix it up nicely. You keep the temperature relatively constant. So that same summer, John Gerhardt came to give a lecture. Now, John Gerhardt is a professor at Berkeley, and he had got very interested in frog oocyte maturation. Now, frog oocytes are big cells, and I'm going to add progesterone, which has been discovered as the key to their maturation. So there goes the progesterone. A dramatic change occurs. A white spot forms. But were you able to see inside, what you would discover that actually that white spot conceals a lot of internal changes, which ends up with a second meiotic spindle forming inside the cell, just in fact underneath the white spot. And what John, who was an expert on enzymology at that time, and not so much of a developmental biologist, pointed out was that a few years before, 8 years previously, Masui and Markert had discovered that what progesterone did was to activate some kind of precursor substance to make a factor which actually catalyzed the G2 to M transition inside these cells. And what was more was that you could show that this factor had a very curious property, namely it turned itself on, because if you sucked a little bit of this cytoplasm out and put it into a fresh oocyte, they underwent maturation very much quicker. You could keep on doing this without the need for any new protein synthesis, DNA synthesis, or RNA synthesis. Clearly something going on inside the egg. And that something was heat sensitive, protease sensitive. Basically, it was an enzyme that catalyzed cell division. And that also was a revelation to me. I thought, gosh, what a wonderful problem. What is this stuff? And the idea that you could have an enzyme that catalyzed cell division struck me as very bizarre. I was used to enzymes like glucose-6-phosphate dehydrogenase, which really didn't do very many interesting things. In fact, most enzymes are extremely dull. And, but an enzyme that catalyzed cell division, wow. And John had been trying to purify it and not succeeding. And actually, he and his colleagues did, however, find some interesting things, namely that it was high in the eggs, and the eggs stayed in this state, this metaphase state, until the sperm arrives and fertilizes them. And then MPF disappears, and then it comes back during the mitotic division. So it's not just for meiosis, also for mitosis. It goes down between meiosis 1 and meiosis 2. And you find it, first of all, in starfish, and then it was eventually found in human cells. And the net result of that was that it became pretty clear that whatever this stuff was, A, you couldn't purify it. If you looked at it, it disappeared. But B, it was kind of universal. So starfish MPF worked in frog eggs, and human MPF worked in frog eggs. I'm not sure that the other way around was ever tried. But that was pretty extraordinary, and it became clear this was it. So I began to wonder a couple of things, namely, did sea urchins have MPF by any chance? And second of all, I don't think I'm going to show you this. No, we were doing experiments with Joan on clams, and when you fertilize a clam egg, it starts making new proteins. So we wondered what these new proteins were, and did they have anything to do with the control of cell division. But we didn't really think about it in a very coherent way, except that there was one important finding, which again we made many, many years before. If you inhibited protein synthesis, the sea urchin eggs did not divide. So clearly one or more proteins made after fertilization was essential for cell division. But again, we weren't thinking very clearly about this. Well, of course, if you inhibit protein synthesis in a HeLa cell, it doesn't divide. But that's because the HeLa cell doesn't grow, and it's not making lots of things. So it didn't, the full logic. And then by 1982, I was getting a bit frustrated with the main problem, namely what turned on protein synthesis. And it frankly wasn't going very well at all. And I read this book by Jacques Lerbe, a Rockefeller scientist, very famous guy, Artificial Parthenogenesis and Fertilization. It turned out that virgin birth was possible in sea urchin eggs. So I sort of began to wonder whether it made a difference whether the sperm was there or not. So one Thursday evening I decided to do a little experiment to see whether the pattern of protein synthesis and parthenogenesis was the same as or different from the pattern of protein synthesis in properly fertilized eggs. And by that time I'd learned how to fertilize the eggs and how to run gels. These were quite important technical advances which I won't go into. So this is kind of molecular theology, really. So here is the Virgin Mary, and she's going to parthenogenetically activate her egg with the Holy Spirit. And one thing I didn't mention about my dad was that both my parents were very religious, and I was brought up very religious. And I can remember clearly at the age of 12 absolutely believing in the reality of virgin birth. I mean, to question it would have been to question the whole floor of my being. I mean, you know, it was an article of faith. I mean, that's important. Anyway, so let's do the experiment. So I did the experiment, very simple experiment. You take some fertilized sea urchin eggs or some parthenogenetically activated sea urchin eggs. When you're sure the fertilized ones are fertilized, you add label, and you just take samples. And you've seen I was used to taking samples at regular intervals to measure rates. And it gave a certain robustness to the experiment. And blow me down, what did I see? Well, you see this protein here? It was the first one you could detect early on, very abundantly synthesized, about 5% of the total. It went up and then it went away. Oh my God, how curious. And then would you believe the luck? So the autoradiogram was barely dry when I ran into John Gerhardt at the wine and cheese party that evening. And John told me about the experiments that he and Mark Kirshner had been doing. And what he told me was that when you triggered this rise of MPF by a starter dose of MPF rather than progesterone, it went up. But for the second rise, you needed protein synthesis. If you added protein synthesis inhibitor, it didn't come back. Now, I'd just seen this result, which I described to him. It's the most exciting scientific conversation I'd ever had in my life. This protein comes and then it goes away again. And then, so clearly, if you inhibit protein synthesis here, it can't come back, and then the cells can't divide. So, I mean, you know, you've suddenly solved the mystery of cell division by doing this one stupid, dumb experiment for the wrong reason. And basically, I mean, that turns out to be right. Again, I went back into Cambridge and told people about this. We did some other experiments. But on the basis of about, 3 or 4 weeks' work, we got a cell paper. The cell paper that said it was wild speculation based on faulty logic, but it was true. And very surprising. And because in those days, you couldn't do this. Ubiquitin had been discovered, but it was discovered in the context of getting rid of misfolded or damaged proteins, not as we now know it as a very well-regulated system that can get rid of any protein you want at any time you want. And we did other experiments, and in the following summers I came back and collaborated with Joan Rudiman, because it turned out that these were the three translationally regulated proteins, and it turned out when we did the experiment that actually there were 2 of these proteins. You can see it much more clearly here. They come and they go, they come back and they come. So this is cyclin A and cyclin B, and this turned out to be ribonucleotide reductase. And the only things that sort of was funny about this was that this is about half an hour, right? Was it really possible you could make enough new enzyme to catalyze cell division in half an hour? And so we did back of the envelope, this is a radioactive band, it's not an actual protein band. And I did, this is about the same concentration as glucose 6-phosphate and nitrogenase or something like that. So, it wasn't very much. So, and then the other problem was that clearly the protein goes up steadily, whereas entry into mitosis is something really rather sudden. So that sort of didn't quite make sense, and that worried us a lot. So I didn't think that what I discovered was MPF, but probably an enzyme that somehow catalyzed the activation of MPF. Well, that's kind of right, as it turned out. But anyway, I'm now in a bit of a problem because I've obviously got a new problem which I could work on, but that's not what I'm supposed to be working on. I'm supposed to be working on the control of protein synthesis. And moreover, I don't know anything about the cell cycle or its control. So what do you do? Well, I decided this was a much, much more interesting problem than the one I had been studying. Moreover, what I had been studying wasn't going very well. It wasn't going very well back home either. We sort of got into a mess. So I thought the best thing is to switch to studying, finding out what cyclin is and what it does. I called it cyclin partly because it cycled and partly as a joke because Tom and I like cycling so much. Well, this was the book at the time, written by Murdoch Mitchison, Paul Nurse and Kim Naismith's advisor. And there was no mention in here about proteolysis. I mean, it simply had never been suggested, even by theoretical biologists, as a theoretical possibility. But as you've seen, I mean, it's absolutely clear it happened. So I started to read about it, and of course everybody knew that the cell cycle had chromosome segregation alternating with DNA replication with gaps in between, and you know, the chromosomes lined up and pulled apart. But what was known about the regulation of this process? Well, in those days, not a lot. I mean, this about sums it up. There was some kind of trigger that triggered mitosis. And even Mazier, the author of this cartoon, said that he didn't think this was a very good idea. But Mark Kirshner and I really liked it, and we wanted to know what this trigger was and whose finger was on that trigger. And that's something actually we still don't know. And I think what this makes clear, you know, lots of things happen when a cell enters mitosis, and a lot of that is reversible, it's complicated. And how the hell do you find out what the starting pistol is? Well, of course, people had made quite a good start on this in the yeast field, and Lee Hartwell was the real pioneer. And what Lee had discovered was that you could make temperature-sensitive mutants of yeast that couldn't do some of these cell cycle transitions. And the one that he and his colleagues particularly focused on was something called CDC28, which controlled these three processes, budding, DNA synthesis, and spindle whole body duplication, which they argued were three such separate biochemical processes. The only possibility was that this must be a regulatory mutation and control a key regulator of the cell cycle. Meanwhile, Paul, who trained as a biochemist and got very bored and discontent with the biochemistry, read Lee's papers and decided he would follow in his footsteps, but use a different yeast, and went to work with Murdoch Mitchison, who worked on this yeast, Schizosaccharomyces pombe, fission yeast, which looks like a giant bacterium. It's about 10 times the size, 10 times the linear dimensions, and could isolate CDC mutants, and when you get a CDC mutant, it can't divide, so they just get longer and longer. And what Paul was trying to do was to enrich for these long mutants by centrifugation, and as a result, accidentally isolated these small cells, because the small cells clump together in big lumps and very rapidly go to the bottom of the tube. And Paul was looking at the cells and realized that at the non-permissive temperature, what happened was that the cell cycle was actually accelerating. So he then embarked on a screen to look for further strains of yeast, different mutations that gave this same Wee phenotype. And he went through, I think, 49 separate isolates which he mapped, and they all turned out to map in the Wee1 gene. But the 50th one was different, and it mapped in a gene that he'd already seen called CDC2. And again, that was a revelation. So Paul argued that CDC2 was a very important regulatory gene because one kind of mutation accelerated cell division, another kind of recessive mutation blocked cell division. So this was a really, really interesting gene. And then it gradually emerged that CDC2 and CDC28 were interchangeable. Molecular biology was beginning to come into vogue, and I know Paul went on a course in Heidelberg to learn how to do it, where he met my friend Richard. After a lot of confusion, it turned out that whereas CDC2 mainly controlled G2N and CDC28 mainly controlled G1S, actually in both these it controls both, and you can either turn it on or turn it off. And when it was was sequenced, it clearly encoded a protein kinase. So I remember walking around a field with Paul in Bath and saying, Tim, it's definitely not a protein kinase because, you know, we make this stuff, we've tested it, and it has absolutely no protein kinase activity. So I said, gosh, you know, if you tried other substrates, oh no, don't need to do that. So actually they've been using casein as a substrate, and CDC2 will never phosphorylate casein under any circumstances. So sometimes not being a biochemist, which Paul certainly isn't, is a bit of a hindrance. And but Melanie Lee in his lab found that CDC2 was present in human cells too, and that was the thing that really sort of made everybody realize that they had been up to this time, about 1988, studying cancer without having the first foggiest clue about how cell division was catalyzed. And then suddenly, when a foot had been put in the door, you know, everybody started working on it because suddenly it became possible and you knew what you had to, you sort of knew what you had to do. But you'll see that cyclin never fit in this, and neither Lee nor Paul had ever isolated cyclin. However, a man called Bob Boer had been sequencing a mutation in POMB called CDC13. And in Mark Kirshner's lab, my friend Andrew Murray had been working on a cell-free system derived from frog eggs, which did cell-free cell cycles in a protein synthesis dependent manner. So we were very much thinking along the lines of, you know, could cyclin, we promised Andrew that as soon as we cloned cyclin, we'd send him the clone and he could test it. So they were very keen, and Mark Solomon in his lab was looking at the sequences of cyclin and trying to isolate cyclins from yeast. So we really believed in cyclins. And Boer came to give this seminar and flashed up his sequence on the screen. And Mark Solomon realized that he was showing the sequence of cyclin. Now, why had Boer not realized that he was sequencing, because sort of the sequence of cyclin was already in the database? The reason was he used the wrong strand. In those days, blast searches only searched with the strand you gave them, and for some reason, the cyclin sequence in the database had been put in the wrong way round. So there was a very funny, I mean, this was extraordinary. So Andrew called me up and said, hey, Tim, you know, guess what? You mustn't tell a cell, and in particular, do not tell under pain of death. Paul knows. CDC13 is cyclin. So as luck or bad luck would have it, the very next week, I was to visit Paul's lab for some reason. So I went to Paul and said, have you been looking at any of your sequences recently? You found cyclin by any chance? And he said, no, I don't think these yeast have cyclin. It turned out that the reason was they were indeed, Ian Hagen was sequencing CDC13, but they were using pirated software. And the pirated software had an amazing, very clever anti-pirating thing built in, which was that every so often it would just completely scramble the contigs. So they weren't getting very good sequence out of the sequencing exercise because of that. I mean, it was absolutely bizarre. But anyway, so I was very disappointed by that because Paul didn't know CDC13 cyclin, and then I had to go away on holiday and I couldn't tell him. But when I came back, the Boer paper was published, and so we could look at the sequence in public, and I said, have you seen this? Cyclin. But by that time it was too late for them. Anyway, so this kind of stuff happens. There is Paul, flanked by, I should have said that at the time, John Pines was the first person to clone sea urchin cyclin B, and Jeremy Minchell was the first person to clone frog cyclins. And that was a real breakthrough because I knew that once we got frog cyclin, obviously humans had cyclins, and I sent John off to Tony Hunter's lab, my old friend and lab mate, to look for the human cyclin, which he very quickly did. Had all the probes and stuff. So I'm working with this pair. I mean, to clone cyclin wasn't trivial because the only assay we had was it was this band on a gel. So we had to use a kind of antisense method. as I say, I was very friendly with Paul. I used to call him to that whenever possible to discuss what's going on. And it turned out that cyclin was the activating subunit of CDC2. And that's really all there was to it. And what's interesting, I think, and Andrew proved that you had to make cyclin in his cell-free system to turn on the CDC2. Very beautiful experiments we won't go into. And I think what was extraordinary was that there were some very clever people working on this problem. And it just never crossed our minds because no protein kinase had previously, only companion subunits to protein kinase that had been discovered were ones that turned the protein kinase off. No one had ever seen a companion subunit that it's just so obvious in retrospect, but at the time we were completely blind to that. And no one thought of it, no one suggested it until it was sort of forced upon us. It was absolutely amazing. So it was really, really simple once you understood. CDC2 was already there. The cyclin got made anew. When there was enough of it, CDC2 turns on and the cell enters mitosis. Then God knows how the cyclin was destroyed. Now we have a pretty good idea. And that turns off the CDC2 because it has no activity. If you don't have cyclin, then the cells exit mitosis. And so, that what had previously been completely mysterious suddenly turned out to be utterly clear and very easily explainable in simple molecular terms. There was still a lot of very important details to be worked out. It's a lot more complicated than that. So that appears to be the end of the story, if you like. But one thing that always bothered me, this was a protein kinase. Kept on discovering protein kinases that control things. So the idea is that in interphase cell, all these proteins have to get phosphorylated. So how many proteins have to get phosphorylated? How much do they have to be phosphorylated? Well, the answer turns out that you need hundreds of proteins to be phosphorylated. There's no one master protein that has to be phosphorylated. And it's very difficult. You have to do experiment on each and every one to find out what difference that phosphorylation makes. Several of them get multiply phosphorylated. And the question is, do you have to have all those phosphates on? So another question is then, at the end of mitosis, if it's true that you got into mitosis by putting all these phosphates on, it must be true that to get back to interphase, you have to take all those phosphates off. And that means that there must be phosphatases. You certainly don't destroy all these proteins and make them anew. Proteins get phosphorylated and dephosphorylated. So this worried me as a biochemist because it's a classic futile cycle. So if you have the kinase and the phosphatase present at the same time, then as quickly as you phosphorylate something, you will get dephosphorylated. So you can never get 100% phosphorylation. So it looked as though there really ought to be a mechanism that when the kinase was on, the phosphatase was off, and when the kinase switched off, the phosphatase came back on. And again, this is another funny thing. It sort of seemed obvious, and yet I never dared to put anybody onto studying this problem, and it's probably just as well. And the point really is, and I hope I've made this absolutely clear, and I think previous speakers made this absolutely clear, that often, it's rather like weather forecasting, it's pretty clear where you have to take the next few steps, but after that, where the road goes, you don't know. There may be no road when you get over the top of the hill, or it may turn left or right, go down. And all you have to do, I tell people say, what's the secret of success in science? I think it's very simple. You put one foot in front of the other and keep going. But you do need to keep your eyes on the horizon. You need to be on the lookout for stuff. And you need to work very hard. I've noticed that some scientists seem to be much luckier than others. And I've noticed that the people who are much luckier than others tend to be the very intelligent ones who work awfully hard. That's how you get lucky.